Okay. Uh, okay. Ask, Shafia. Okay. Before continuing, everyone of you quickly answer the question. Everyone of you answer the given question. Everyone. Slowly, man. Others. What about others? Quickly. Good. So, if you see this, it's a simple only, soya bean, which belongs to the leguminous plant, that is by rhizobium. Casuarina will be the Frankia, is it? And Gunnera will be the nostack. Azolla will be the Anabina, right? What is your question? No. Chemoattractants are releasing from the plant, Nandini, that they are going to activate the NOD-D gene, which is present in the bacteria. Okay. All right. Okay. Now, okay. Let me continue for 30 minutes. Don't disturb in the middle. Listen to me very carefully. Okay. If every one of you very quickly recall, we have completed the first step of the nitrogen metabolism. That is one of the most important and the biggest step. What was that? Nitrogen fixation, is it? Now, after fixing the nitrogen, now we fixed the nitrogen into what? We fixed the nitrogen into NH3 or NH4. That is what we fixed the nitrogen. But I told to all of you very clearly, NH3, NH4 also not simple forms. We can't assimilate them very easily. So, very shortly, we have to reduce them into more simple forms, such as NO2 as well as NO3. Don't get confused with that. So now let's focus on the nitrification. It's such a simple step it is. Again, the question was asked in the exam. You have to remember them. What is the nitrification? Conversion of ammonium ions to the nitrates. So where there is a, focus all of you, where there is a conversion of NH3 or NH4 plus into NO2 and NO3. I have very clearly told you this entire process is said to be the nitrification only, but what is different? The bacteria involved is entirely different, right? Here the bacteria we generally call it as, I kept it here, nitrosomonas. Correct, na? And you can take nitrobacter. This is the nitrobacter. And I'll give you another example, nitrosomonas and nitrosococcus. Don't get confused, friends. One is called nitrosococcus and another one, we called it as the nitrococcus. There's no point of getting confused here, okay? Nitrosomonas and nitrosococcus. Friends, this is a nitrosococcus and it is a nitrococcus. Don't get confused. So, now finally, what we have done, we have converted NH3 or NH4 to NO2, then followed by the NO3. Now the point is very clear. Because now I told you, right, the first biggest step is the nitrogen fixation. The second very biggest, the second biggest step is the assimilation. So far, we converted N2 to NH3, NH4, NO2, NO3. Finally, we have to assimilate it. So now the question is very clear. What happens if I don't assimilate? What is the question? What happens if I don't assimilate the NH3, NH4, NO2, and NO3? What happens? Undoubtedly, friends, since they are the nitrogen derived compounds, they will cause the highest level of the toxicity in the plant as well as in the animal. So that is the reason you should not store any kind of a component in the any kind of a nitrogen derived component in the plant body. So what is the only way? Tell me. The only way is I can convert all of them into amino acids. This is the only thing that I can do it. Now, that is what exactly I can call it as the assimilation, incorporation, utilization, making use of it. Let's see how. Every student listen. What happens if you store the nitrate? What happens if you store the ammonium? I am telling you in a very simple words. Nitrate, if you store, every student remember, nitrate plant can store. The reason is that nitrate cannot show a highest harmful effect in the plant. Remember my words very, very correctly. But you understand. So this is a plant stored the nitrate. It won't show any kind of adverse effect on the plant growth. But you remember there is a livestock. Suppose example, a herbivore like a cow or a buffalo or a goat, which is eating that particular plant. Do you really think that they can digest this particular nitrate? No. Correct, na? So once this particular herbivore consume that particular nitrate in their liver, what happened? Nitrate is getting reduced to the nitrite. Then what happened? That nitrite will go and combined with the hemoglobin, which causes methamoglobinemia. I can tell you in simple words, friends. Nitrate and nitrite, both we generally call it as a potent carcinogenic. What is the meaning of that? At extremely lower doses also, friends, they can cause the cancer activity. Don't get confused with that point. That is the nitrate. So that's the reason it is very harmful that we should not store the nitrates and nitrites. Now tell me, what happens if the ammonia stores? I'll tell you in a simple lines to all of you. Friends, if the ammonium stores, I'll tell you in a simple words, it will block entire electron transport chain machinery in the mitochondria also, in the chloroplast also. The ammonium can destroy all the photosystems in the plant. So it will block the electron transport chain in the mitochondria as well as on the thylakoid membrane. That's why ammonium also should not store. Now, but the interesting point is, how does, everyone of you focus, how does this particular ammonium can destroy all this? Because these are all present on the membrane. How does the ammonium can enter up to the membrane level? That's a good question. Now, everyone remember very correctly. This is the stroma, matrix, or the cytoplasm. All of you focus. NH4, which is the ammonium. At the stroma, we all know that, friends, the pH is extremely higher. NH4 combined with the OH. Then what is going to happen, friends? It is going to release the NH3. It is converted into NH3. Now, I told you multiple times to all of you, NH3 can easily cross the plasma membrane with the help of a aquaporins. So NH3 cross the membrane. Inside the lumen, lumen is always acidic pH. Again, NH3 combined with H plus and converted into ammonium, then block entire machinery. So what I'm trying to explain you is, if the nitrate and ammonium, if they present enough, they will cause the toxicity most of the cases. That is the reason it is very important to convert nitrate and ammonium into the most simple usable forms. What are those usable forms? Undoubtedly, the usable forms are amino acids only. Let's focus on that. See, I'm requesting you keep reading the statements, whichever is there in the slides. That is more important for the exam. Okay. Now, all of you listen. I want to absorb the nitrate also, nitrite also, ammonium also. I want to convert everything into the amino acids. Now, all the students listen, every one of you. Nitrogen is usually available in the soil as the nitrate ion. This is also one of the statement that was asked in the exam. What is the question? Nitrogen usually available in the soil. It is in the form of what ion? Nitrate ion. Now, plant cannot make any kind of a amino acid directly from the nitrate, because what is the question? Why plant cannot make any kind of amino acids directly from the nitrate? Because nitrate is not a utilizable form. Now you tell me what the plant has to do. It has to convert that nitrate into ammonia, because if ammonia is present, we have an amino group. So by using the amino group, amino acids can be synthesized. Now you tell me what the plants are going to do now. First, plants are going to absorb that particular nitrate present in the soil. Later, they are converting that particular nitrate into ammonium. Later, it is getting converted into amino acid. Now, the question is crystal clear. How does this particular nitrate can be absorbed into the soil? That is important here. From the soil into the root hair via H plus NO3 symporter. This is also they will raise the question. Now here itself I am asking you a question. Okay. Now I want all the students has to think properly and answer. Okay. Tell me SSV. According to this given question, my question is very simple. Nitrate absorption follows primary active transport. Is this statement is a true or false statement? Swati, Ritika, Bhavya, everyone of you. My question is, nitrate absorption from the soil into the plant, it is primary active transport. Absolutely wrong statement, right? It is obviously following the secondary active transport. We all studied multiple times, because here we are not using the ATP directly. The proton pump is pumping the H plus, and that H plus is helping, or it is creating the gradient in such a way that NO3 also can come in the same direction. No need to get confused with that. That is true. That is true. Good, good. So now let's focus. Now you know what I'm going to explain you. I'm going to explain you first how does the NO3 converted into NO2, how does the NO2 converted into NH4, and I will explain you how does the NH4 converted into amino acid. And friends, what is the best way of remembering all together? Like hormones, we all people has to focus on comparison. If you compare one enzyme with another, you can remember it very, very easily. So, and it is extremely important for the exam to remember the location. Okay, location is also very important, friends. Incorporation of nitrates into organic compounds like amino acid proteins, which we call assimilation. Every one of you focus. First, I am converting NO3 to NO2. So I will be keep on telling you the simple way. Follow me. Okay, first step. What is the first enzyme we all studied, friends? We studied a first enzyme, very important enzyme, nitrogenase enzyme. Now, what is the enzyme I'm going to read? I am converting what to what? I'm converting nitrate into nitrite. So friends, I am reducing something. What I am reducing? I am reducing NO3. So the enzyme involved is undoubtedly nitrate reductase enzyme. Okay, NO3 to NO2. So I am reducing the nitrate. So the enzyme is nitrate reductase. And what is very important for the exam? Where this particular enzyme is present. That is crucial here. Nitrate reductase enzyme is present within the cytosol. That's a very important statement. Okay, now keep on comparing. Let's compare once again. Let's compare the nitrogenase enzyme structure. If all the students quickly recall, nitrogenase enzyme contain dinitrogenase and dinitrogenase reductase. Now let's look into the nitrate reductase enzyme. Every point is important. It composes of two identical subunits, two. So that is important, friends. So it's a dimer. It's a two identical subunits. Every subunit contain three prosthetic groups. That's a crucial point. What are those three prosthetic group? One is FAD, another one is heme, no need of structure. Third one is called molybdenum, associated with a organic compound called terrin. Again, my friends, they will ask a straightforward question. What is the question they can ask? Which, now every student answer me. Which cofactor is commonly observed in the nitrogenase enzyme and nitrate reductase enzyme? Undoubtedly, what was that? Molybdenum, which is the cofactor which is commonly observed in both enzymes. So again, quickly recall, who is the electron donor for the nitrogenase enzyme? We all studied very clearly that, friends, the electron donor for the nitrogenase enzyme is ferredoxin. I hope you all people remember it. Now, who is the electron donor for the nitrate reductase enzyme? That is important for the exam again, which is the NADH. Now, again, some students will get confused. Sir, is it a NADPH or is it a NADH? In some books, it was written that it is only NADH. But in the Lincoln Tays and other some standard reference book and some research journals also, it was written that NADH. So in the exam, if they give NADH also or NADPH also, don't get confused. Both are acting like a electron donor only. So requesting all of you not to get confused with that. Okay, now listen. What's happening now? NADH is donating the electron to FAD, FAD to heme to molybdenum contain terrin, that to the NO3. Finally, NO3 is getting reduced to what, friends? NO2. Now, I am asking you a very simple question to all the students. My question is, let's consider there is a deficiency of the molybdenum in the plant. Now, tell me what happens. My question is, there will be a deficiency of a molybdenum in the plant. What happens? No reduction. Very good. What is the meaning of that? Which means that nitrate is getting not assimilated in the plant. It is getting stored in the plant. So storage of the nitrate, obviously it causes the carcinogenic effect. So molybdenum deficiency leads to the accumulation of the nitrate in the plants. Don't get confused with that. So what we have done, we have finally converted NO3, which is the toxic form, to the NO2. But to be specific, compared to the NO3, NO2 is more toxic. The meaning of toxic is more reactive, means it can easily react with the many cells in the body. And especially it combines with the liver cells also. That's why it is important we should not store the nitrate also. Sorry. Now what we have to do with this particular nitrite? We have to convert this nitrite into ammonium. Why we have to convert into ammonium? Because ammonium having the amino group, which is required for the synthesis of amino acids. That's why our ultimate goal is to convert into ammonium. Again, important question for the exam. Where is the site of the location of the nitrate reductase enzyme? It is exclusively present in the cytosol. But now the point is that we produced nitrite. Where we produced nitrite? In the cytosol only. But you know what happens? This nitrite from the cytosol, remember everyone, it is going to enter into the chloroplast and the plastids. What is the reason why the nitrite has to travel from the cytosol into the chloroplast and plastids? What is the reason? Because the presence of an enzyme which is called nitrite reductase enzyme, which is only specifically present in these organelles. So NO2 is entering into the chloroplast of leaves and plastids in roots. There NO2 is reducing to the NH4. Again, once again, all of you quickly recall, who is the electron donor for the nitrogenase enzyme? Ferredoxin. Who is the electron donor for the nitrate reductase enzyme? that is NADH or NADPH. Now, who is the electron donor for the nitrite reductase? Again, ferredoxinol. Again, ferredoxin. No need to get confused. Now, friends, look into the picture. If all of you quickly recall, nitrate reductase contains what? 2 identical subunits and 3 prosthetic groups. But if you look into the nitrite reductase, you only look into the picture, look into this. There will be only 2 prosthetic groups are present. You can see it very clearly. What does it contain? Iron sulfur cluster and only 2 prosthetic groups and 1 heme. And again, who is the electron donor? Very important one. Electron donor is ferredoxin. So ferredoxin donate the electrons to the iron sulfur cluster to the heme to the NO2, and finally NO2 is getting reduced to the ammonia. Now finally what we have done? We reduced NO3 to NO2, we reduced NO2 to ammonia, because that is only our ultimate goal. Why only ammonia? Because ammonia is the one which will be helpful in converting into amino acid because of the presence of a amino group. Simple point. Now, again, some students are asked me a question that, sir, when ammonia is readily available, why only glutamine is synthesizing first? Why glutamate is synthesizing for second? Why proline amino acid is not synthesizing? Why asparagine is not synthesizing? See, it is depends on the presence of the enzymes in that particular location. Okay. No. It is not called ammonification. Listen. I'm telling you, right? It is called assimilation. Listen. I'll explain in the upcoming slides. Now, why only we are synthesizing glutamine or glutamate is, it is depending on the availability of that particular enzyme. So this ammonia, we are converting into glutamine amino acid and we are converting into glutamate amino acid. This is also one of the questions that was asked in the MP set exam. Okay, focus all of you. I have already told you many times to all of you friends, synthetase is the enzyme always ATP dependent. When you read the name synthase, it is not dependent on the ATP. Maybe it is dependent on the other energy precursors such as NADH or FADH2 or whatever it may be. Now listen. So all the students listen. Is it necessary to remember the cofactors? I already told you friends, it is mandatory to remember the cofactors. Never ever neglect the cofactors. Now you listen. All the students listen. There are 2 enzymes I have taken into the consideration. One is called glutamine synthetase enzyme, glutamate synthase enzyme. What is the function of glutamine synthetase? It synthesizes the glutamine. What is the function of glutamate synthase? It synthesizes the glutamate. Now let's talk about first enzyme. What is the name of the first enzyme? Glutamine synthetase. So it is involved in the synthesis of glutamine. Now all the students follow me. If I want to synthesize the glutamine, what I am using? I am using the glutamate first. I hope every student is getting my point. In order to synthesize the glutamine, I am using the glutamate first. Every one of you focus on the diagram. So this is the glutamate, and you tell me what is the differences between the glutamate and glutamine. All the students focus on the given diagram. What's the structural differences between the glutamate and glutamine? Glutamate having only 1 amino group, glutamine having the extra amino group. Now the question is that, who is giving this extra amino group to the glutamate? That is the question. Ammonium. So ammonium will donate the amino group to the glutamate. Upon receiving the amino group, the glutamate is getting converted into glutamine. So since it is a synthetase enzyme, undoubtedly it is exclusively dependent on what, friends? ATP molecule. No need to get confused with that. You have to remember the cofactors. Mandatory. Now what's the second enzyme, friends? Second enzyme is what? Glutamate synthase. I am synthesizing glutamate now. Now all of you focus on it. So again, this is also very important question asked in the exam. Glutamine synthetase, it is located in the cytosol also. It is located in the chloroplast also. Big friends, this is also asked statement I have seen in the some 2012 or 13 question paper. So it's very important. You have to remember the locations. Remembering locations is mandatory. Now next one, glutamate synthase, also called as a GOGAT. Now what's the function of GOGAT or glutamate synthase? As the name itself suggesting, it is involved in the synthesis of glutamate, right? It is involved in the synthesis of glutamate. Now focus. Now see this interesting step. Every student follow this interesting step. Just now what we discussed, in the previous step, we have synthesized the glutamine. Is it? How did we synthesize the glutamine? We have taken the glutamate, we combined with ammonium. Correct now? We have taken ammonium, we combined with glutamate, we donated this amino group, we converted into glutamine. Now, to that glutamine, I am just adding the 2-oxoglutarate. That's all. Upon addition of the 2-oxoglutarate to the glutamine, which is going to give rise to the 2-glutamate molecules. In simple words, by using one glutamate, every student follow me. By using one glutamate, by using glutamine synthetase enzyme, glutamate synthase enzyme, finally we produced 2 glutamates. Don't need to focus on the structures, don't waste the time. Now, the glutamate synthase enzyme, which is also called as a GOGAT. What's the, so you understand, synthetase used the ATP. Now what the synthase will use? Either NADH or the FD. Again, this is also question. There are 2 type of GOGATs present in the plant. One GOGAT which uses NADH, one GOGAT which uses FD. And what is important for the exam? Where they are located. That is important. NADH GOGAT is always present in the non-photosynthetic tissues, whereas FD GOGAT exclusively present in the photosynthetic tissues. That's the only fundamental difference you have to remember. Now, let me tell you about a specialized enzyme. Right now, we have used glutamine synthetase. We produce 1 glutamine. We use glutamate synthase. We produce 2 glutamate. Now, there is a specific enzyme. We call it as, one second, glutamate dehydrogenase enzyme. So again, they'll ask a question in the exam. Which of the following enzyme can be used in the reversible reaction of synthesis of glutamate? You see this. You remember, first in the previous step, I combined ammonia with the glutamate. I produced glutamine. Again, I combined glutamine with 2-oxoglutarate. I produced 2 glutamate. Now, I am not doing any of this. What I am doing, directly I am taking the ammonium. I am combining the ammonium directly with 2-oxoglutarate. Ammonium combined with the 2-oxoglutarate. What's happening now? This ammonium is donating the amino group. That's all. To this double bond. Hence, there is a breakdown of the double bond. Then this 2-oxoglutarate converted into glutamate. But plant won't prefer this actually. Why it won't prefer this actually? Because if we use the GOGAT, enzyme, we are getting 2 glutamates. But if we use the glutamate dehydrogenase enzyme, we are getting only 1 glutamate. That's why, remember every one of you, glutamate dehydrogenase enzyme is not preferred when compared to the GOGAT. Suppose example, if the GOGAT is deficient in the plant, compulsory plant has to use the GDH. And again, all the students has to understand my statements. Just now I told you that there are 2 type of GOGAT. One GOGAT is NADH dependent, one GOGAT is FD dependent. Similarly, friends, there are 2 type of GDH. One GDH exclusively dependent on the NADH, another GDH exclusively depends on the NADPH. That's also very crucial. The The GDH, which is dependent on the NADH, found in the mitochondria. And the NADPH, it is found in the chloroplast. Trust me, this kind of statements are very common. So friends, finally, what we have done? We converted the ammonium into amino acid successfully. Now what happens? Now, one amino acid can be converted into another amino acid. How is this possible? It is possible by a mechanism called as transamination. As the name itself very clearly explaining to us, I am just transferring the amino acid. What I am doing? Transferring the amino acid. Now you see this, transferring the amino group. Now all the students follow me. We have produced the glutamate. Now, by using a glutamate, I want to produce the aspartate. How can I produce the aspartate from the glutamate? By using another versatile enzyme called as aspartate amino transferase enzyme. What it is actually doing? You only look into this. Glutamate contain what? Amino group. Correct, na? So now, and this is what? Oxaloacetate. Now, just try to understand. Now, this glutamate combined with the oxaloacetate. Then glutamate is converted into the aspartate. Then oxaloacetate is getting reduced to the 2-oxoglutarate by an enzyme called as amino transferase enzyme. That's a simple one. You no need to focus on a single structure also. Just remember the name of the enzyme. So next, glutamine I have converted into asparagine. How is this possible? Again, the amino group of the glutamine shifted to the aspartate. Upon receiving this amino group, aspartate converted into asparagine. Upon donating the amino group, glutamine converted into glutamate. So in this way, just by the transfer of the amino group, one amino acid can be converted into another amino acid. Again, one of the statement that they asked in the exam. Tell me, Shafia, what is the name of the cofactor that is essentially involved in all transamination reactions? Mauli, Vasuki? What's the name of a cofactor that is essentially involved in all the transamination? Every one of you compulsory remember that, which is the pyridoxal phosphate, which is the vitamin B6. I want all of you has to remember this compulsory. Okay. Now all of you please come back to our major slide. If every one of you quickly recall what happened to this? 1 second, 1 second, just 1 second. Correct. Okay. Vitamin B6. Now, so far, what we studied, friends, every one of you focus. Someone asked about ammonification. Follow me. So now, nitrogen fixation is done. Nitrification is done. Assimilation is also done. Correct, na? Who assimilated? Undoubtedly, plants assimilated. There's no doubt about it. Is it? Now, friends, from the plants, who will take how animals will get the amino acids, proteins just by eating the plants. By eating the plants, we animals will get everything. That's absolutely fine. Now, what is the condition when the plant die? What is the condition when the animal die? Listen, every one of you. When the plant die and animal dies, what happens? All the dead organic matter can be converted into organic. I can tell you simple words: proteins, amino acids, nucleic acid, which are organic, converted into inorganic such as carbon, hydrogen, oxygen, nitrogen. Now what the bacteria will do? Bacteria starts using this particular nitrogen, because bacteria also require the nitrogen compulsory. So it uses the nitrogen for its growth and for its development. But the point is very clear. Here there are highest amount of the nitrogen is present here. But bacteria can't utilize that much amount of the nitrogen. How much ever the nitrogen required for its growth and development, it will use only that much amount of the nitrogen. What about the excess nitrogen? It is going to fix in the soil again in the form of ammonia. This step only we call it as ammonification. So in a simple words, ammonification is a process of fixing the excess nitrogen in the soil, that is called ammonification. Now you only look into this. Now plant benefited, animal benefited, bacteria also benefited. But actually from where we have taken the atmosphere, taken the nitrogen, we taken the nitrogen from atmosphere. So what we have to do now? We have to send back that particular nitrogen to the atmosphere. By that, it will complete the nitrogen cycle. Just a minute. And that process only we generally call it as a denitrification. Again, what you all people have to remember for the exam. Remember for the exam is examples. So we are sending NO3. NO3 back to the atmospheric nitrogen and NO2 gases. You can see it, right? You can see now we are converting nitrates back to the atmospheric nitrogen. No need to get confused with that. And here I have mentioned the extra examples. I am requesting all the students to remember the extra examples. Focus on the examples without fail. Now, but I told to all of you a specialized type of process and a specialized bacteria, which we can call anammox. All the students follow me. Why we have given the name anammox? ANA represents anaerobic ammonium oxidation. What was the full form? Anaerobic ammonium oxidation. So as I already told to all the students, there are many bacteria are not involved in this process. Hardly 5 bacteria are involved, which are converting NO2 and NH4 directly to the nitrogen. So what are those 5? You people has to remember. If you did not find the time, there's no problem. But only remember these 5 names: Brocadia, Cuninia, Anamoxoglobus, Zetinia, Scalnuda. Okay, if they give the question, that it based on our luck. Okay, so these 5 are the bacteria which show the mechanism of anammox, where ammonia and nitrate are biologically converted into the nitrogen gas. Please don't get confused with that. Okay, all of you answer the question. Take your time and properly answer the question. So fast, man. Everyone is so fast. This fast also will, okay, try it. Just now we studied, no? Okay, you people can eliminate this option very, very easily, right? Maria, look into this. One of the prosthetic group attached to the terrain is what? It is not magnesium, right? It is a molybdenum. Correct, na? So undoubtedly, yeah. So this statement will be the wrong statement. Here only you can eliminate it. Then only you can come to the conclusion. Nitrate reductase of higher plants is composed of 2 identical subunits. One subunit of nitrate reductase contains 3 prosthetic groups. A prosthetic group attached to the both subunit is heme. Absolutely true, right? Answer is number 1. Okay. Take your time. I want all of you to give the proper answer for this. Take your time. Choose the correct statements. Use the elimination. You are just reading the options. Use the elimination. Okay. First of all, every student tell me, what about the option 4? Option D is correct or wrong? Tell me, everyone. Every student tell me option D is a correct option or not. Undoubtedly, option D is a correct answer. Then you eliminate the option 1 also. You eliminate the option 2 also. What is there in that? Here only we got conclusion, right? Option D is the correct answer. Now you tell me. Option C, the metal associated with the enzyme nitrogenase enzyme. What is the metal? Every one of you, what is that metal? What is that metal? The metal is the molybdenum. Correct, na? Hence, this will be also undoubtedly wrong answer. Hence, you can come to a conclusion. Just the A and D. Okay. I hope it's clear for everyone, the nitrogen metabolism. Is it clear, everyone? The entire nitrogen metabolism? Quickly. Okay. See, okay. Now, plant physiology, I will go little faster only, but when it comes to the molecular biology, I will teach little slower. I want you to become a super duper experts in the molecular biology and unit 4. Okay, now listen, in the tomorrow class, we have only 3 hours of time. In the 3 hours of time, I have to complete 3 hormones. So now I am focusing on the very important hormone, auxin, another important hormone, gibberellic acid, another important hormone, ethylene. But if I find the time, very surely I will try to cover the another hormone. So again, I'm telling to all of you, it is more in the soil only compared to the ocean, Ritika. That's a good question. More in the soil only. Okay. Now, all of you listen. If you are belongs to the OBC, ST, SC, I am only telling you very confidently, this crash course itself sufficient. But if you are belong to a general category, you have to read the double of these people are doing. It's not like about the category. It is about 1 mark or 2 marks difference. That 1 mark or 2 marks also, it's very important here. So now what I am trying to explain you is the general category people, try to read more compared to these people, because here if they get 100 to 102 also, they are getting the JRF, but here you people has to get the 120. Hence, you people has to read beyond the crash course, beyond the revision course. Whatever I am focusing is the most important one, but make sure that you read more. But as I promised, yeah, it's not the beyond PPT level, Ritika. It's beyond the concept. It's not about the book. No. I'm telling more topics. Suppose example, tomorrow I'm teaching only 3 topics, 3 hormones. You people has to read another 4th and 5th hormone also compulsory, because you should not take the chance. Right. Now, very important one. In the tomorrow, I will do the 3 hormones and you have to solve the questions. And I'm telling you, the people still who are having difficulty in the molecular biology, biochem, developmental, evolution, and methods in biology, unit 13. I will cover 90% of the topics very fastly, okay, very effectively, very specifically towards the exam point of view. So you focus on that. Okay, for revision, this is sufficient. Okay, for revision. Okay. All right. I'll see you in the tomorrow class. And remember, I have observed that only 6 people have given me the answer. Friends, please don't neglect. It is not like a motivation class. You have to take the super responsibility and you have to submit the answer. You no need to tell me your score. Just to tell that you have did the test. That is more important. Within 15 minutes, I will send you the questions of the nitrogen metabolism specifically on that topic. Do it mandatorily. Okay. Do not neglect that. Okay. And what is the doubt, Shafia? Okay. 1 second. I'll quickly explain that. What's the doubt? Okay. No problem. No need to send me. Just tell the marks enough. Okay. I mean to say, just tell that you did it. What is the first line defense mechanism in photooxidative damage? Is it a photochemical quenching? Now everyone tell her doubt. Shafia is saying that in case of a photoprotective mechanism, the first mechanism is the photochemical quenching. Is it a true or a false statement? In a photoprotective mechanism, the first defense mechanism is photochemical quenching. Undoubtedly. Correct, na? So you see this, you remember, Shafia, the first is we are leaving the excess amount of the photons in the form of a heat. So the releasing in the form of a heat only photochemical quenching. Correct, na? So the statement is absolutely true statement. No need to get confused with that. All right, all right. Okay. So I'll see you in the tomorrow class. Let's continue. But I'm telling you, friends, for you people, trust me, this 50 days, I will irritate all of you more until you come to the track. I will take the class on Sunday also if it's needed. I'm telling you, I will take the class if it's needed at 5:30 a.m. also. I will take it for very sure if it's needed. Okay. Every one of you be prepared for that and work very, very hard this 50 days. Okay. Work extremely harder. Okay. And do not give up it in the middle at all. Okay. Now please don't give it too much importance to your college work, your household work. Give the super importance here. Okay. No, why I am saying is before ending, I'm telling you one statement. Keep this point in your mind. There is someone in India, okay, preparing 10 times harder than all of you to crack the exam. So it is all about competition. So you can survive in the competitive exam only when you spend the time. It is not your intelligence. It is only hard work. Okay. So spend more time on preparation and remember it. Okay. All right. I'm sending you the questions within 15 minutes. Everyone do it for the tomorrow class without fail. All right. All right. Thank you, all of you. Thank you so much. Thank you.